Watson and Crick found the DNA structure to be a double helix with two antiparallel polymer strands consisting of linked nucleotides. The major structural components of DNA are a deoxyribose sugar backbone, nitrogenous bases, which are the information carriers, and a linking phosphodiester bonds that are responsible for DNA's negative charge. DNA stands for deoxyribonucleic acid, which means that in the chemical structure we see a hydrogen without an oxygen present on the 2' carbon. There is an importance to the carbons present on the ribose ring. The 2' carbon tells us if it is DNA or RNA based on if there is an oxygen present with the hydrogen attached here. The 1' position carbon is where the base is attached. The 3' position carbon will join with the 5' carbon of the phosphate connected to the next nucleotide. The 5' carbon, which is above the ribose ring, attaches to the previous nucleotide's phosphate group. The nucleotides are linked in a directional manner due to the base pairing in charge of the phosphodiester bonds interacting with the charge of the nucleotides, which is why we read DNA in a 5' to 3' direction. There are two kinds of nitrogenous bases: pyrimidines, which are thymine, cytosine, and uracil, which is only found in RNA. And there are purines, which is adenine and guanine. Adenine pairs with thymine using 2 hydrogen bonds, and cytosine pairs with guanine using 3 hydrogen bonds. Hydrogen bonds are weak though, so these are not the main force joining the two strands. Base pairing is seen where adenine can only bind with thymine and guanine can only bind with cytosine. The two strands of DNA are opposite of each other, including opposite nucleotide base pairing and the opposite direction of the 5' to 3' end, creating an anti-parallel structure. This, in addition to the negative charge of the phosphate backbone and the base stacking of the hydrophobic nucleotide bases in the center of the strand, causes the two anti-parallel strands to create the helical shape. Handedness describes the direction that the helix is turning. If the spirals point up and to the left, it is left-handed. If the spiral points up and to the right, it is right-handed. The normal form of DNA is right-handed. The helical shape shows minor grooves and major grooves, which alternate. The major grooves are good for proteins that come and interact with the bases present to help unwind the structure for transcription. DNA's negatively charged hydrophilic phosphate backbone is neutralized by the presence of salt cations, which shield the negatively charged phosphates, ultimately eliminating the repulsive force between the two anti-parallel strands. The salt increases the DNA's melting temperature, making it harder to denature the DNA molecule.